Hi everyone, my name is Nancy Espinosa and I will present the following word about the proposal we have to generate a skin protective agent based on encapsulated Carvacrol. The human skin is the outer covering of the body. That's why it is constantly exposed to external aggression such as pollution and ultraviolet radiation. The overexposed to UV rays induce the formation of free radicals, which are highly reactive and very unstable species that affect cellular components such as DNA, proteins, and lipids, and leading to many skin disorders include premature aging and pathologies like cancer. Skin cancer is one of the most important diseases due to its increasing incidence in recent years. According to recent statistics, every year around the world, about 300,000 cases of melanoma and around 1 million cases of non-melanoma are diagnosed. Some of the strategies to prevent skin cancer are using sunscreen, wear clothes, hats, and glasses, limit the time in the sun, and recently, the use of exogenous antioxidant has been proposed to reduce the level of damage. Nowadays, there is a growing interest in using products of natural origin, such as essential oils, which are complex mixture of compounds extracted from plants. They have commercial significance in the agronomy, pharmaceutical, and cosmetic industry thanks to their beneficial biology properties. Essential oils contain around 20 to 60 compounds, from which carvacrol is one of them. Carvacrol is a phenolic monoterpenoid found mainly in the essential oils of oregano and thyme. This component has received special attention for its biology properties, mainly for its antioxidant activity. Carvacrol is a highly volatile compound with a hydrophobic nature and instability under to environmental factors. These chemical characteristics limit the use of carvacrol. Therefore, its encapsulation in polymeric nanoparticles has been proposed to provide protection, control release, and maintain its biological activity on the skin. Nanoparticles are defined as nanosized solids which can be made of synthetic or natural materials and act as a release system for active compounds. Some of these advantages of incorporating biological active compounds in nanoparticles are increasing their stability, controlling their release, and improving their distribution on the skin. However, the administration of active compounds in nanoparticles needs to be truly evaluated to determine that the formulation applied are safe, non-toxic, and do not affect the physiological parameters of the skin in order to maintain the integrity of the skin barrier after application of the formulation. The general objective of this work was to evaluate the effect of carvacrol-loaded nanoparticles on the biophysical properties on the skin in order to establish its potential use as a skin protective agent. The general strategy consisted of 4 stages. First, a suspension of carvacrol-loaded nanoparticles was obtained and it was characterized physiochemically. In the second stage, a chromatographic method was developed and validated for the quantification of carvacrol from the nanoparticles. Subsequently, the antioxidant activity of encapsulated carvacrol was evaluated. Finally, biological tests were carried on to determine the effect of the formulation on the surface of the skin. The elaboration of the nanoparticles was carried out by the nanoprecipitation method. The polymer and the carvacrol dissolved in a water-miscible solvent were added into an aqueous solution. The addition of the organic phase into the aqueous phase under magnetic stirring caused interfacial deposition of the polymer, leading to the formation of nanoparticles. Subsequently, an evaporation process was carried out to eliminate the solvent to obtain the nanoparticles in the aqueous phase, which were used in the following assays. For the physicochemical characterization of the nanoparticles, the particle size, polydispersity index, and the zeta potential were determined using a Zeta-Sizer for Malmer instrument. The carvacrol nanoparticles show a size less than 200 nanometers. According to previous research, this particle size allows a varied distribution of nanoparticles on the skin. Additionally, a polydispersity index of 0.109 was obtained. This parameter is associated with the homogeneity of the dispersion of the nanoparticles. A value lower than 0.200 indicates a homogeneous system, Therefore, the polydispersity index obtained in this research indicates that the most of the nanoparticles have similar sizes. This is important because a uniform particle size distribution will allow the individual interaction of the nanoparticles to occur uniformly on the skin. For the zeta potential, a value of minus 8.8 millivolts was obtained. This parameter is related to the stability of the nanoparticles. Also, the magnitude of the zeta potential of the Carvacrol nanoparticles was not high. It's enough to provide stability to the formulation, which did not show change in the size polydispersity index and zeta potential. Also, visual modifications such as separation phases were not shown for at least 4 months. Additionally, the charge density is also related with the interaction with the biological system, which means that the nanoparticles with negative charge contribute to the deposition of the skin. Continue with the chemical characterization of the nanoparticles. They were evaluated by infrared spectroscopy to determine if there were unexpected interaction between the compounds used for the formulation of nanoparticles. Bands belonging to the hydroxyl group were observed in the spectrum of Carvacrol, as well as peaks corresponding to metal and metallite groups. In the spectrum of the polycaprolactone, that was the polymer used for the formulation of the nanoparticles, bands belonging to the carbonyl group were observed. In the spectrum of the twin, that was the surfactant used as a stabilizer in the formulation, peaks corresponding to hydroxyl and carbonyl groups and characteristic bands of the ester were observed. In relation to the spectrum of the Carvacrol nanoparticles, the bands of the functional groups observed in the spectrum of each compound of the formulation were detected. No additional bands corresponding to the formation of new bonds were observed, which indicates that there is only chemical interaction between the compounds used for the formation of the nanoparticles. As part of the physicochemical characterization of the nanoparticles, the amount of Carvacrol incorporated in the nanoparticle was quantified. First, an analytical method by gas chromatography was developed and validated in order to obtain reliable results. To validate the system, a calibration curve was constructed with a Carvacrol standard in a concentration rate of 25 to 400 micrograms per milliliter. Subsequently, the injection was carried out in the gas chromatograph under this optimal condition, and finally, the corresponding chromatograms were obtained. For the validation of the system, the parameters of linearity, detection limit, quantification limit, and accuracy were evaluated. To verify the method, Carvacrol nanoparticles were analyzed. They were subjected to an extraction process, and a calibration curve was prepared in a concentration range of 25 to 400 micrograms per milliliter. For the validation of the method, the accuracy and precision parameters were evaluated. The results of the validation of the analytical method are shown below. An R squared than 0.99 was obtained. The limit of detection and quantification were lower than the lowest concentration of the working interval. Meanwhile, accuracy values were less than 10%. On the other hand, in the validation method, relative standard deviation values lower than 10% and recovery percentages between 70 and 110% were obtained. All the parameters evaluated in the validation comply with what is established by the Secretaría de Salud and the International Council for Harmonization, will ensure reliable results in the quantification of the Carvacrol incorporating the nanoparticle. Following with the quantification of Carvacrol, the Carvacrol nanoparticles were subject to successive extraction with methanol and analyzed using the previously validated gas chromatography method. The percentage of encapsulation obtained was 13%, and the percentage of encapsulate efficiency was 28%. These results allow to determine the amount of encapsulated Carvacrol, which is essential for a correct dosage of the active, which is closely related with the effect on the skin and the antioxidant activity. Stage 3 involved the measurement of the antioxidant activity of the encapsulated Carvacrol by means of DPPH and ABTS tests. Initially, the mean inhibitory concentration of a Carvacrol standard were obtained from both methods. These reference values were compared with those obtained from the Carvacrol-loaded nanoparticle suspension. ABTS and DPPH methods showed an IC50 of 4.76 ppm and 183.79 respectively. The percentages of inhibition of the complete suspension of Carvacrol-loaded nanoparticles and the supernatant of Carvacrol-loaded nanoparticle suspension were determined, obtaining percentage of inhibition of 90% for the ABTS test and 38% for DPPH test. It should be mentioned that uncharged nanoparticles were also evaluated, as well as the supernatant of uncharged nanoparticles. They didn't show antioxidant activity in any of the tests carried out. Likewise, the antioxidant activity of the carvacrol extracted directly from the nanoparticles pellet was determined by means of the DPPH test, obtaining an inhibition of 54%, which indicates that the carvacrol that is encapsulated have an antioxidant activity once released. These results show that the formulation has an antioxidant activity that could protect the skin against free radicals. In this work, an ex vivo model of porcine skin in frank diffusion cells was used to evaluate the permanence of carvacrol in the stratocorneum after being in contact with the carvacrol-loaded nanoparticles for 4 hours. A tape stripping technique was performed. This technique consists of the successive removal of the layers of the stratocorneum using a tape. The carvacrol present in each tape was extracted with methanol and quantified by the previously validated gas chromatography method. The amount of carvacrol in the stratocorneum was 42 micrograms of carvacrol at a depth of 6 micrometers. After exposure in frank diffusion cells of the carvacrol-loaded nanoparticles and the nanoparticles without carvacrol during 1, 2, and 4 hours, the effects on the skin barrier function were evaluated by measuring the loss transepidermal water, the pH, and the amount of stratocorneal water. The graph showed the results of water loss. This parameter is used to analyze the homeostasis of the skin barrier, where the increase in the values indicates more water loss, which means a damage to the skin barrier. The graph shows that there was no significant difference in the water loss values after exposure of the nanoparticles without carvacrol for 1, 2, and 4 hours with respect to the baseline value. This indicates that the nanoparticle without carvacrol doesn't disturb the function of the skin barrier. On the other hand, it is observed that there was a significant difference in the water loss values after application of the carvacrol-loaded nanoparticles at 2 hours of exposure, with respect to the baseline value. However, after 4 hours of exposure of the carvacrol-loaded nanoparticles, no change were observed in the water loss values with respect to the baseline value. Therefore, it could be assumed that the carvacrol-loaded nanoparticles temporarily modifies the water loss values. This figure shows the result of skin hydration, which is another indicator of epidermal function. The graph shows that when both formulations are applied to the surface of the skin for 1, 2, and 4 hours, the skin presents a statistically significant increase in the water content of the stratocorneum compared to the baseline values. These results indicate that both formulations improve the hydration of the stratocorneum. This figure shows the results of pH, which is also an important parameter to evaluate the homeostasis of the skin. The graph showed that there was no significant difference in the pH values of the skin after the exposure of both formulations for 1, 2, and 4 hours compared to the baseline value. These results show that the both formulations preserve the pH of the skin and do not contribute to the alteration of the skin homeostasis. The conclusion of this work are: Carvacrol-loaded nanoparticles were prepared using the nanoprecipitation technique and show optimal physicochemical characteristics which contribute to the deposition and subsequent release on the skin. The formulation maintained the antioxidant activity of carvacrol after contact with the skin. The biophysical evaluation of the skin after the application of the carvacrol-loaded nanoparticles for 1, 2, and 4 hours showed that the formulation increased the hydration levels preserves the pH values and doesn't change the water loss values, which together suggest that it doesn't contribute to the skin barrier damage, representing an alternative as a skin protective agent. Thanks for your attention.